Assalamu alaikum students. We already studied that how a nerve impulse travels from nerve terminal to muscle membrane, and that is through neuromuscular transmission. Now we will see that how muscle action potential spreads and how it brings about muscle contraction, which is the ultimate function of muscle to bring about movement. Resting membrane potential of muscle fiber ranges from minus 80 millivolts to minus 90 millivolts, while the duration of action potential is around 1 to 5 milliseconds. Velocity of conduction is usually from 3 to 5 meters per second. Now, let me briefly tell you the mechanism of skeletal muscle contraction. As I have already told you that an action potential that is in the nerve fiber, the action potential in the nerve fiber that leads to the secretion of acetylcholine and the opening of acetylcholine gated channels. And this opening, it results in depolarization of muscle membrane, which causes the sarcoplasmic reticulum to release the calcium ions. This release of the calcium ions results in contraction. And in the end, for muscle relaxation, there is pumping back of the calcium ions into the sarcoplasmic reticulum back. So, I told you, I will tell you how muscle action potential spreads. To understand this, it is important to learn about the transverse tubules sarcoplasmic reticulum system. Transverse tubules, uh, and wonder why they are called transverse tubules, because they lie transverse to the longitudinal axis of muscle fiber. So these transverse tubules or the T tubules, they originate from the sarcolemma. The transfer tubules are open to the exterior of the muscle fiber. The transverse tubules or the T tubules, they communicate with extracellular fluid surrounding the muscle fiber, and they penetrate all the way through the muscle fiber from one side of the fiber to the other. And their function is to conduct the action potential from sarcolemma to the interior of the muscle fiber. Now, sarcoplasmic reticulum is composed of two major parts: large chambers, which are called terminal cisterns, and the longitudinal tubules that surround all the surfaces of the actual contracting myofibrils, plus an active calcium ion pump that is also present in the membrane of sarcoplasmic reticulum. Well, in short, the spread of the action potential to the interior of the muscle fiber, it is achieved by the transmission of the action potentials along the transverse tubules, or you can say T tubules. When the T tubule it receives the action potentials, there is a release of the calcium ions inside the muscle fiber cells, and these calcium ions they cause contraction, and this overall process is called excitation contraction coupling. So if you are asked to explain excitation contraction coupling, in the first step the action potential travels along the muscle fiber membrane in the same way the action potentials they travel along the nerve fiber membranes. And next, the action potential depolarizes the muscle membrane. Well, third step is the release of calcium ions by the sarcoplasmic reticulum. As one of the special features of the sarcoplasmic reticulum is that it contains calcium ions in high concentration. Now, these calcium ions are released when an action potential occurs in the adjacent T tubule. Then the calcium ions they bind with the troponin C, causing the sliding of actin filaments on myosin filaments and finally leading to muscle contraction. The role of calcium in that once the calcium ions they have been released from the sarcoplasmic tubules, muscle contraction continues as long as the calcium ions remain high in the sarcoplasm. And as I already told you, that a calcium pump is located in the walls of sarcoplasmic reticulum. Now this pump, it pumps the calcium ions back into the sarcoplasmic reticulum. Uh, and keep in mind that the normal resting state concentration of the calcium ions, which is around 10 raised to power minus 7 moles. Now, this normal resting state concentration in the cytosol, which bathes the myofibrils, this is too little to elicit the contraction. So you get it that how in the normal resting conditions the usual concentration of the calcium ions this is not able to cause the contraction, and to get the contraction the release of the calcium ions from the sarcoplasmic reticulum is necessary. This sliding filament theory explains that contraction occurs by the sliding of filaments. This sliding is caused by the forces which are generated by the interaction of cross bridges from the myosin filaments with the actin filaments. So as a result, the sarcomeres they shorten in length and the Z lines they move close together. Let's now look at the inhibition and the activation of the actin filaments. Well, in this figure, you can see the actin filament, which is in an inhibited state. And actin filaments, they remain inhibited in the relaxed state of muscle. The active sites on the actin filaments of the relaxed muscle, they are inhibited, so these sites, they cannot attach to the heads of myosin filaments to cause contraction. Now, here you can see that how the actin filament that is activated, because when calcium ion, which is released from the sarcoplasmic reticulum, it combines with the troponin C. The location of troponin C, it changes because this part of the troponin molecule, it becomes heavy with the attachment of the calcium ion, so it displaces, and along with the displacement of troponin C, tropomyosin molecule also is displaced. The tropomyosin molecule, which earlier was covering the active sites on the actin filament, now it has moved into the groove between two actin filament strands. So the active sites, they have been exposed. Since the active sites, they get exposed, so they interact with the head of the myosin filament and they bring about the contraction. Mm, this walk-along theory of skeletal muscle contraction, it explains the interaction between the activated actin filaments and the myosin cross bridges. The sliding filament theory states that the heads of the cross bridges, they bend back and forth, and they step by step walk along the actin filament. As this figure, it shows the heads of two cross bridges attaching to and then disengaging from the active sites of an actin filament. All right, here you can see the term power stroke. Power stroke is the tilting of myosin head after it attaches with an active site, and this tilt, it causes to slide the filaments towards the center of sarcomere. Well, here you can see the steps of cross bridge cycling while I'm trying to make this topic interesting for you. Uh, and this figure is also showing the energy dynamics during the muscle contraction. There are certain factors affecting muscle performance. Skeletal muscle performs better when it is regularly exercised, for the reason that with the regular aerobic exercise, skeletal muscles are trained to improve their ability to use oxygen. Muscles also improve in their efficiency by which they use energy sources such as fats. Anaerobic training such as weightlifting. It increases the muscle bulk because it increases the size of individual fibers within the muscle. The amount of actin and myosin filament overlap. Now, this overlap, it determines the tension which is developed by the contracting muscle. I repeat again that the tension which is developed in the muscle, that is dependent on the amount of the actin and myosin filament overlap. Now, what is the relation of muscle length to the tension? You see, this relation of the muscle length to the tension, it demonstrates that maximum contraction, it occurs when there is maximum overlap between the actin filaments and the myosin filaments. The greater the number of the cross bridges pulling the actin filaments, the greater will be the strength of the contraction. Now we move on to some of the contractile properties of skeletal muscles. Uh, you must know that larger muscle fibers, they produce more force than the smaller fibers, because the large fibers, they contain more actin and myosin. So more myosin cross bridges, they are available, which result in more force. Now, muscle tone is the natural firmness of muscles when they are not, when even when they are not flexed. And keep in mind that some fibers, they are always contracted. There are two types of muscle contractions: isometric and isotonic. While most the activities, they include both isotonic and the isometric contractions. Well, let's start with the isometric contractions, where you see the word metro, it means to measure, uh, especially measure the length. So, in isometric contractions, in the isometric type of muscle contraction, there is seen no decrease in the muscle length. I repeat, there is no decrease in the muscle length in isometric type of contractions, but the muscle tension increases. Although keep in mind that true isometric contractions, they cannot be generated in the intact body. Now, the isometric contractions, they are basically important for maintaining posture and for supporting the objects in a fixed position. Although isometric contractions, they do not result in body movements, but energy is still expended. Here you can see the mechanism of the isometric contraction. You see, in this type of contraction, there is no change in the length of the muscle fiber. Do you think that no work is being done? The work is still being done as the tension in the muscle that changes. So, energy is also consumed. Examples of isometric contractions, they include pushing a very heavy table, then the movements involved in bodybuilding, or you can give the example of holding a book steady using an outstretched arm, and standing. standing still, sitting, and even posture maintenance. These are the examples of the isometric contractions. Now we move on to the isotonic contractions. In this type of the muscle contraction, muscle length decreases, but the muscle tension remains same. And the examples of isotonic contractions, these include such as lifting an object, walking, or moving any part of the body. Functions which require the energy, ATP, during contraction, they are to activate the walk-along mechanism, uh, and the active transport of calcium ions back into the sarcoplasmic reticulum to terminate the contraction. So keep in mind that energy is required not only for the contraction of muscle but also for the relaxation of muscle. Regarding the sources of energy, you see the baseline concentration of ATP in the fiber that is sufficient to maintain full contraction for only 1 to 2 seconds. And later, new energy, new ATP that is generated from certain sources, and these sources they include the phosphocreatine compound present in the muscle, and then there is glycolysis. You see, there is glycolysis and there is also the oxidative metabolism, but glycolysis, it is 2.5 times more rapid than the oxidative metabolism. However, 95% contribution of the energy that is achieved by the oxidative metabolism of carbohydrates, fats, and proteins. Hmm. Types of skeletal muscle fibers, they include the classification which categorize them into fast and slow muscle fibers. And this classification is based on the biochemical capacities of the muscle fibers. Two main differences among these fiber types, they are basically the speed of contraction as the name is indicating, and also the type of enzymatic machinery which is used to energize the contraction. And keep in mind that every muscle of the body, it is composed of a mixture of so-called fast and slow muscle fibers. Let me now give you brief detail about these muscle fibers. Slow muscle fibers, they have comparatively lower myosin ATPase activity, resulting in slower twitches, in slower contractions of these fibers. And these slow muscle fibers, they are also called type 1 fibers, or you can call them red muscle. Now, these slow muscles, they have got smaller fibers, more extensive vascular system, and their resistance to fatigue is high. Slow muscle fibers have got greatly increased numbers of mitochondria to support the high levels of oxidative metabolism. And examples of slow muscle fibers are soleus and the muscles of back. Well, next are the fast fibers. These fast muscle fibers, they are also called type 2 fibers or the white muscle fibers. These muscles, they have got large fibers for great strength of contraction, and they have got extensive sarcoplasmic reticulum for the rapid release of calcium ions, and they have also got large amounts of glycolytic enzymes for the rapid release of energy. Then the fast fibers, they have less extensive blood supply, fewer mitochondria, because the oxidative metabolism here, in the case of fast fibers, this oxidative metabolism, this is of secondary importance. And the fast fibers, they also have a deficit of myoglobin, and that is why they are white in appearance. Now, the examples of fast fibers, they are tibialis anterior, or you can say the extraocular muscles.